According to NCERT syllabus, in second year biology, the second topic is sexual reproduction in flowering plants. Before going to that, let's discuss reproduction. Already we discussed about reproduction in the first chapter in biology. Reproduction is a vital process. Reproduction is a vital process. It is an important biological process. And without reproduction, species cannot exist for longer time on the planet of Earth. If there is no reproduction, there is no continuity of species from one generation to another generation. That means without reproduction, there is no existence of species for longer time on the planet of Earth. And during reproduction, a new individual is produces, either by asexual reproduction or by sexual reproduction. A new individual or new offsprings or new children are produces during by sexual reproduction or by asexual reproduction. If an offsprings are produces by asexual reproduction, there is no variations in the offsprings. But if an offsprings are produces by sexual reproduction, a new variations are created. So, sexual reproduction creates a new variations in offsprings. Offsprings are produced by either by asexual reproduction or by sexual reproduction, but variations created in the offsprings only by sexual reproduction. During sexual reproduction, the diploid sporophytic cells transformed into haploid gametes, male gametes and female gametes. For example, this is one male parent body. The male parent body dominant phase is sporophyte. Sporophytic ploid is diploid. Female parent also diploid region is the dominant stage in female parent body is sporophyte. The sporophytic cells in the male parent transformed into haploid male gametes by the process of meiosis or male gametogenesis. The process of male gametes from the sporophytic cells of male parent by meiotic cell division is called male gametogenesis. Then the sporophytic cells transformed into haploid gametes. These are male gametes. This is diploid female parent sporophytic cells. Whenever it is undergoes meiosis, then it produces haploid female gametes by female gametogenesis. After male gametogenesis and female gametogenesis, the diploid sporophytic cells transformed into haploid male gametes and female gametes. These haploid male gametes and haploid female gametes physically contact to each other by the process of fertilization or syngamy to form diploid zygote. Now this diploid zygote in higher plants or higher organism, it is undergoes mitotic cell division or mitosis to form diploid embryo by the process of embryogenesis. During embryogenesis, there are two events can take place. One is cell division, another one is cell differentiation. Now this diploid embryo can transformed into or differentiated into a new organism by the process of organogenesis. All these events, male gametogenesis and female gametogenesis, fertilization and embryogenesis and production of seeds and production of new organisms. All these events can take place in specialized organ in a flowering plants or in angiosperms is called as flower. All these events, gametogenesis, fertilization, embryo development, all the events in flowering plants or in angiosperms can take place in a specialized modified organ is called as flower. Let's we discuss about flower. Generally, the study of flowers is called as anthology. Once again I repeat, the study of flower is called as anthology. Production of flowers is called as anthesis. The study of flowers is called as anthology. Production of flowers is called as anthesis. Flower is a modified structure. It is the main site for sexual reproduction in flowering plants or angiosperms. Generally, flower is a fascinating organ. Flower is a fascinating organ. You can find different colors and different types and different shapes of flowers in the diverse of living world. The flower is a fascinating organ. It is a modified and condensed shoot. Flower is a fascinating organ. Flower is a modified and condensed shoot system. Generally, how flowers are formed in an angiosperms or in flowering plants. Generally, during vegetative phase in animals, it is called as juvenile phase. During vegetative phase in plants, the shoot apical meristem. During vegetative phase in plants, the shoot apical meristem, the shoot tip having shoot apical meristem, it is actually dividing tissue. When the juvenile vegetative phase stops, when the reproductive phase will be starts in a flowering plants in angiosperms, the shoot apical meristem modified into a reproductive meristem. The reproductive meristem is transformed into floral primordium or floral axis, inflorescence axis. These floral primordium can produce as buds. Buds can produce as flowers. This is the process of formation of flowers in the plant body before the initiation of sexual reproduction in flowering plants or in angiosperm. After formation of this flower, the process of production of these flowers is called as anthesis. The study of these flowers is called as anthology. Flower is a fascinating organ. Flower is a modified and condensed shoot system. Flower is the main site for sexual reproduction in angiosperms or flowering plant. Flower is the main site for sexual reproduction in angiosperms or flowering plants. Generally, the flowers are having different objects. Generally, flowers we are used for beauty of appreciation. That is called as aesthetic. This is the first object of flower. And these flowers are generally we are used for different cultural activities. And flowers are having religious in nature. And flowers are having social object. Generally, flowers we are used as ornamental. These are the objects of flower. The objects of flower, aesthetic and cultural and religious and social and ornamental uses. Now we discuss about structure of bisexual flower. In the structure of bisexual flower, the stalk of the flower is called as pedicel. The top of the pedicel having a flattened structure is called as thalamus. Thalamus is also called as receptacle, is also called as torus. On the thalamus, there are 4 modified floral appendages or floral organs are present, such as outermost floral appendages in a bisexual flower is called calyx. The second floral appendages is called corolla. The third floral appendages is called androecium. The fourth floral appendages is called gynoecium. Out of these 4 whorls or circles in bisexual flower on the thalamus is classified into 2 groups. The first group is non-essential whorls. The non-essential whorls is also called as helping whorls, or it is also called as accessory whorls. The third and fourth whorls are called as essential whorls. The essential whorls is also called as reproductive whorls. Essential whorls is also called as reproductive whorls. Out of 4 modified floral appendages or organs on the thalamus, classified into 2 groups. The first group is non-essential whorls or helping whorls or accessory whorls, such as calyx and corolla. The second one is essential whorls or reproductive whorls. Let's we discuss in a bisexual flower, the outermost whorl is called as a calyx. Generally, calyx is green in color. Its function is photosynthesis and protection during bud condition of flower. The units of these calyx is called as sepals. Let's come back to second whorl or second circle of bisexual flower is corolla. The units of this corolla is called as petals. These are different colors and different sizes and different shapes due to presence of anthocyanin pigment in the vacuoles of petals. It attracts insects for cross pollination. These are not having any capability to participate in sexual reproduction, then calyx and corolla is called as non-essential whorls or helping whorls or accessory whorls. And let's come back to the third circle or whorl of bisexual flower is called as androecium. The units of androecium is called as stamens. These stamens can responsible for formation of male gametes during sexual reproduction. So that's why androecium is a male sex organ. During sexual reproduction, male gametes are produced from the third whorl of bisexual flower. The innermost whorl or fourth whorl of bisexual flower is called as gynoecium. The units of this gynoecium is called as pistil, is also called as carpel. It is a female sex organ responsible for formation of female gametes for sexual reproduction. So the androecium and gynoecium responsible for formation of male and female gametes, these are help for sexual reproduction. Then third and fourth whorls are called as essential whorls or reproductive whorls. Now in this class we discuss pre-fertilization structures and events in a flowering plants. Now we discuss pre-fertilization structures and events in flowering plants or angiosperms. Male gametogenesis and female gametogenesis means male gamete formation and female gamete formation and pollination. These are the 3 events comes under pre-fertilization. The first pre-fertilization event in sexual reproduction in flowering plant, now we discuss. In a bisexual flower, the third whorl of bisexual flower is called as androecium. Androecium is a male sex organ. The units of androecium is called as stamens. One of the stamen from bisexual flower, if I draw here, each stamen having 2 main parts. One is filament, another one is anther. Filament is a long and slender stalk-like structure. The long and slender stalk-like structure in a stamen is called as filament. And the top of the filament or distal part of the filament having a fertile structure, it is called as anther. Most of the anthers are bilobed. Most of the anthers are bilobed. It has two lobes. Each lobe, it is separated by running a longitudinal structure, and its form bilobed anthers. So, each lobe having two thecas. These two thecas are separated by a longitudinal sterile vasculated structure is called as connective tissue, simply we called as connective. If a connective tissue is present in anther, then the anther is two lobes, that is two thecas, dithecous anther. In monothecous anther, the connective tissue is absent, it has only one theca. Most of the anthers are bilobed and dithecous because region is a sterile vasculated longitudinal tissue is called as connective tissue. And one more important characteristic feature, this is proximal part of stamen. The proximal part of stamen attaches to thalamus and petals. This is very most important point. The proximal part of stamen or filament of stamen attaches to thalamus and petals. The distal part of filament in stamen having a fertile structure is called as anthers. Generally, anthers is of two types, dithecous anthers and monothecous anther. In dithecous anther, a sterile connective tissue is present, then it is from two thecas. But whereas in monothecous anther, sterile connective tissue is absent, the number of thecas in an anther is 1. Most of the anthers are bilobed anthers. This is one lobe and this is one lobe. So, the angiospermic anthers are bilobed and dithecous anthers. For example, this is dithecous anther. In the structure of dithecous anther, a longitudinal sterile connective tissue is present, is called as connective tissue. Then the number of thecas are 2. With the help of sharp blade, if we made a transfer section of dithecous anther under microscope, the structure of dithecous anther or young anther anatomy looks like this. The transfer section of young dithecous anther under microscope, it looks like this. The transfer section of young anther or TS of young anther or anatomy of young anther. Each anther at 4 corners having 4 microsporangias. This is one microsporangia, this is one microsporangia, this is one microsporangia, this is one microsporangia. The transfer section of young anther or anatomy of young anther at 4 corners, 4 microsporangias are present or 4 pollen sacs are present. Each pollen sac or microsporangia surrounded by 4 anther wall layers are present. Outermost anther wall layer is called as epidermis. The second anther wall layer is called as endothecium. The third anther wall layers is called as middle layers. The innermost anther wall layer is called as tapetum. Once again, I will discuss the transfer section of young anther or anatomy of young anther under microscope, the appearance of diagrammatic presentation like this. In the anatomy of dithecous anther, the 4 corners dithecous anther under microscope having 4 microsporangias in 2 lobes. This is sterile connective tissue. Connective tissue is present in Dithecus anthers, absent in Monothecus anthers. Whenever the connective tissue is present, this anther is having two thecas. This is one theca and this is one theca due to presence of connective tissue. Each theca having two microsporangias. The Dithecus anther having 4 microsporangias. So that's what is called as tetrasporangiates. Tetrasporangiate. Dithecus anthers is also called as tetrasporangiates. Each microsporangia or pollen sac surrounded by 4 anther wall layers. Outermost anther wall layer is called as epidermis. The second anther wall layer is called as endothecium. The third anther wall layer is called as middleus. The innermost anther layer is called as tapetum. These 3 anther wall layers, epidermis, endothecium, middleus, its main function is protection and dehiscence. Dehiscence means breaking at mature condition of anther for releasing of microspores or pollen grains. The innermost anther wall layer is called as tapetum. Its main function is nutrition or nourishment to growing sporogenous tissue present inside the pollen sac or microsporangia. Each microsporangia having a mass of sporogenous tissue, generally it's a diploid, is called as a sporogenous tissue. The beginning or growth of the beginning development of sporogenous tissue for nourishment, the nutrition given by the innermost layer of anther wall is called as a tapetum. Tapetum is the innermost layer of anther wall. It's nourishing the growing sporogenous tissue in a pollen sac or microsporangia. For easy to discuss, for cellular organization, if you made clear cut differentiation of each and every cell in anther walls under compound microscope at one sector of transfer section of Dithecus anther, it looks like this. For cellular organization, we made a sector plan under compound microscope. So at one sector plan in Dithecus anther, outermost layer is called as epidermis. It is the first layer of anther wall. It is a single layer. Outermost layer of anther wall is called as epidermis. It is a single layer. Its function is protection. At mature condition, it's responsible for dehiscence or breaking. In some plants, like exceptionally, the name of plant is called Arcithobium. In the plant of Arcithobium, outside the epidermis, one more epidermal like structure is produces. This is called as exothecium. Exothecium is one more epidermal layer is formed in one plant is called as Arcithobium exceptionally. And inner to epidermis, the second layer of anther wall is called as endothecium. So inner to inside endothecium, there is a different cellulosic bands are present. These are called as alpha cellulosic bands. Alpha cellulosic bands from the tangential walls of inner walls, from the tangential inner walls of endothecium, many alpha cellulosic bands or microfibrils are produced. Due to presence of these microfibrils or cellulosic bands, it retains the water and loses the water. That means endothecium having hygroscopic in nature. Okay. And it is also responsible for protection and dehiscence of anther at mature condition. In the plants of Hydrocaritaceae, in the family members of Hydrocaritaceae family members, alpha cellulosic bands are absent. Hygroscopic nature is absent. Inner to endothecium, the third anther wall layers in Dithecus anther anatomy is called as middle layers. The number of middle layers ranges 2 to 3 in number. Inner to endothecium, the third layer of anther wall is called as middle layers. The ranges of number of middle layers is 2 to 3 layers. It is also main function is protection at mature condition. It is responsible for dehiscence, for release of pollen grains from the pollen sac. Inner to middle layers. The fourth anther wall layer is called as tapetum. The innermost layer of anther wall is called as tapetum. Then compared to epidermal cells, endothelial cells, middle layer cells, the cells of tapetum is bigger in size. First characteristic feature. Generally, in epidermal cells, each cell having one nucleus. In endothecium, each cell having one nucleus. In middle layers, each cell having one nucleus. But in tapetal cell, each cell having either one nucleus or more than one nucleus. This is second characteristic features of tapetum. Once again, innermost layer of anther wall is called as tapetum. Tapetal cells are bigger in size, it has more than one nucleus and it has dense cytoplasm and it has more nutrients. Due to presence of dense cytoplasm and nutrients, it is giving nourishment or nutrition to growing sporogenous tissue. So that's why this tissue is called as nourishing tissue or nutritive tissue. Whenever the sporogenous tissue, diploid sporogenous tissue present inside the pollen sac or microsporangia, it is getting food material from the tapetum. This sporogenous tissue develops into microspore mother cell. These microspore mother cell undergoes meiosis, can produce 4 haploid microspores. This phenomenon is called as microsporogenesis. Once again, I recall first pre-fertilization events during sexual reproduction in flowering plants. The first pre-fertilization structures and events is stamens. Stamen is a male reproductive unit. Each stamen having 2 parts, filament and anther. Filament is sterile. The proximal part of this filament of stamen attaches to thalamus or petals. The distal part of this filament having a fertile part is called anthers. If a connective tissue is present, the anthers are dithecous. If the connective tissue is absent, the anthers are monothecous. Most of the anthers are bilobed anthers. And each anther lobe having 2 pollen sacs, each anther theca having 2 pollen sacs, dithecous anther having 4 pollen sacs, tetrasporangia. If we made the transfer section or anatomy of dithecous anther or young dithecous anther under microscope, it looks like this. The 4 corners of transfer section of these young anther having 4 microsporangias are pollen sacs. Each microsporangia or pollen sac surrounded by 4 anther wall layers. Outermost anther wall layer is called as epidermis, second anther wall layer is called endothecium, third anther wall layer is called as middle layers, innermost anther wall layers is called as tapetum. In the sector plan of this transfer section of Dithecus anther under compound microscope, the cellular organization like this. The outermost layer of one part of transfer section of young Dithecus anther, outermost layer is called as epidermis. It is a single layer. Its function is protection. At mature condition, it is responsible for dehiscence, for release of pollen grain. The second layer of anther wall is called endothecium. Endothecium also function is protection. At mature condition, it is responsible for dehiscence. The inner tangential walls of endothecium having alpha cellulosic microfibrils, it's retain the water and loses the water, and it's having hygroscopic in nature. Inner to endothecium, the third anther wall layers is called as a middle layers. The ranges of number of middle layers 2 to 3 in number. It is also function is protection and dehiscence or breaking at mature condition of anther. Inner to middle layers, the innermost layer of anther wall or fourth layer of anther wall is called tapetum. Then compared to epidermal cells, endothelial cells, middle layer cells, the size of tapetal cells are bigger. The number of nucleus, one or more than one nucleus, having dense cytoplasm, it's giving nourishment or nutrition to growing sporogenous tissue inside the microsporangium or pollen sac. Generally, the tapetal cells are bigger in size, having more than one nucleus with dense cytoplasm. This tapetum generally is of two types: glandular tapetum and amoeboid tapetum. If a tapetum is glandular or secretory in function, it secretes sporopollenin, pollen kits and compatible proteins. Sporopollenin, compatible proteins, pollen kits produced by which of the following type of tapetum means secretory tapetum or glandular tapetum. If a tapetum is amoeboid type, second type of tapetum, the tapetal cells are ruptured and it's giving nourishment to growing sporogenous tissue. In the beginning of this sporogenous tissue, getting nourishment from the tapetal cells and develops into diploid microspore mother cell. These diploid microspore mother cell undergoes meiosis to produce 4 haploid microspores or pollen grains. This phenomenon is called as microsporogenesis. So, the topic is microsporogenesis. It is one of the pre-fertilization event in this chapter. Before going to microsporogenesis, once again just we recall anatomy of transfer section of Dithecus anther under compound microscope. In the transfer section of young anther or anatomy of young anther, each pollen sac or microsporangia having diploid sporogenous tissue. This pollen sac or microsporangia surrounded by 4 anther wall layers. Outermost anther wall layer is called epidermis. Second anther wall layer is called endothecium. Third anther wall layer is called middle layers. Fourth anther wall layer is called tapetum. Generally, tapetal cells are bigger cells than the other anther wall cells. These are binucleated, polyploidy condition, having dense cytoplasm, and it is also called as a nutritive layer or innermost layer of Dithecus anther. It is nourishes to or it supplies nutrition to or nourishing to growing sporogenous tissue. The innermost layer of anther wall tapetum is of two types: glandular tapetum or secretory tapetum. Second one is amoeboid tapetum. The glandular type of tapetum main function is help for secretion of a hard organic substances is called sporopollenin, and it is responsible for production of pollen kits, group of pollen, and it is responsible for production of compatible proteins. All these structures to form Ubish bodies. The second type of tapetum is amoeboid type of tapetum. When the amoeboid tapetum is ruptures, the most cytoplasm in amoeboid type of tapetum, it's help for nourishing to growing sporogenous tissue. When the diploid sporogenous tissue in microsporangium or pollen sac getting food material from the tapetum, it develops into sporogenous tissue, develops into microspore mother cell or pollen mother cell. Now this pollen mother cell or microspore mother cell is diploid. Reason is it's directly developed from diploid spore mother cells. Now this diploid microspore mother cell MMC, pollen mother cell PMC undergoes meiotic cell division, then it produces 4 haploid microspores or microspore tetrad. This phenomenon is called microsporogenesis. Once again, I repeat the definition for microsporogenesis. The process of formation of 4 haploid microspores from diploid microspore mother cell or pollen mother cell by meiotic cell division is called microsporogenesis. The process of microsporogenesis can take place inside the pollen sac or inside the microsporangium. The process of microspore tetrad formation during microsporogenesis is of 2 types. The first one is successive type, another one is simultaneous type. For example, in successive type of pollen tetrad formation or microspore tetrad formation generally we are seen in monocotyledon plants or monocots. In this type of pollen tetrad formation, the diploid microspore mother cell undergoes meiosis to form pollen dyad, then it is undergoes meiosis to form pollen tetrad. This method of cytokinesis is called as successive cytokinesis. It is the most advanced type of cytokinesis during microsporogenesis. It is seen in monocots or monocotyledon plants. This is also called as isobilateral tetrad. The second type of pollen tetrad or microspore tetrad formation is simultaneous type of tetrad formation. This method we are seeing in dicotyledons. In this, the diploid microspore mother cell or pollen mother cell undergoes meiosis, then it produces dyad of pollen grains. Now this 2 pollen dyad, then it's formed tetrad of pollens. This method is called as simultaneous type of cytokinesis. Out of these 2 types of cytokinesis or pollen tetrad formation, successive or isobilateral tetrad formation is most advanced, seen in monocots. At mature condition of anther, these tetrad of pollen grains or microspores separate to each other, and each microspore or each pollen grain can develop into male gametophyte. During microsporogenesis, tetrad of microspores or pollen grains are formed. Before going to that, a new concept, let's we discuss once again the definition for microsporogenesis. Generally, it is a diploid microspore mother cell or pollen mother cell. The diploid microspore mother cell or pollen mother cell undergoes meiosis. It produces 4 haploid pollen grains or microspore. The formation of tetrad of microspores or tetrad of pollen grains from diploid microspore mother cell or pollen mother cell by meiosis is called as microsporogenesis. Here I am giving definition. The process of formation of 4 haploid microspores or pollen grains from a diploid microspore mother cell or pollen mother cell by meiotic cell division is called as microsporogenesis. And after microsporogenesis, thousands of microspores or pollen grains are formed in pollen sac or microsporangium. At mature condition, this tetrad of microspores or pollen grains separate to each other, and one of the microspore or pollen grain structure under microscope we discuss now. Now the class is structure of pollen grain is also called as microspore. Generally, the study of pollen grains is called as palynology. This is the first point about pollen grain. The study of pollen grains or microspores is called as palynology. Second one, microspores are the beginning cell. Pollen grains or microspores are the beginning cell or starting cell for male gametophyte. Pollen grains is also called as the microspores or the beginning cell or the initial cell for male gametophyte development. Clear? The study of pollen grains is called as palynology. Each pollen grains, it indicates beginning or starting of male gametophyte development. Each pollen grains, it looks like spherical shape or round shape. Each pollen grains, it measures 25 to 50 millimicrons diameter. Once again, I repeat, each pollen grain is a spherical shape or round shape. Each pollen grains, it measures 25 to 50 micrometers diameter. The size of pollen grains is not constant for all angiosperms or flowering plants. It varies from one pollen grain to another pollen grain. Some of the plants are having smallest pollen grain, some of the plants are having largest size of pollen grains. We discuss later. Each pollen grain, it is surrounded by 2 unit membranes. These are called as sporoderms. Each pollen grains, it is surrounded by 2 unit membranes is called as sporoderms. Outer sporoderm or outer pollen grain wall layer is called as exine. Inner pollen grain wall layer is called as intine. Exine is very hard and thick, having different designings. Reason is the exine is chemically made up of a hard organic substances. The name of hard organic substances is called as sporopollenin. Already we discussed in the transfer section of young anther, innermost layer is called as the tapetum. Tapetum secretes sporopollenin. That sporopollenin is an hard chemical substances. It is present in the exine of pollen grain. Once again, each pollen grain is made up of 2 unit membranes, exine and intine. Exine is very hard and thick, having different designings. Reason is the exine is chemically made up of a hard organic substances is called as sporopollenin. That sporopollenin secreted by tapetum of transfer section of diathecous anther wall. This sporopollenin, it's very hard chemical substances. Due to presence of sporopollenin in exine, the exine is resistant to strong acids and high temperature and strong alkali. No enzymes for known is not able to degrade sporopollen substances in enzyme. Due to presence of sporopollen is a chemical organic substances present in exine of pollen grain. The pollen grains are resistant to strong acids and high temperature and strong alkali. No enzymes are not able to digest this sporopollen. That's the reason these pollen grains are preserved more years. That means it is responsible for fossilization. Fossilization means for more period of time pollen grains are preserved due to presence of hard chemical substances sporopollen in the exine of pollen grain. The fossilization of pollen grains is happened due to presence of sporopollen in the exine of pollen grains. And one more. At some areas in the exine of pollen grain, a thin areas are present or apertures are present. These structures are called as germ pores. The germ pores are present in the exine of pollen grain. Where the sporopollen chemical substances is absent in the exine of pollen grain, those areas are thin areas. The thin areas are called as germ pores. And come back to inner to exine. The second sporoderm or pollen grain wall layer is called as intine. Intine is very smooth. Region is it's chemically made up of two substances, cellulose and pectin. It is also called as pectocellulose. Due to presence of smooth substances like pectin and cellulose, the intine is very smooth. And each pollen grain is single cell, unicellular, having single nucleus, uninucleate, having dense cytoplasm and vacuoles. Due to presence of different designings in the exine of pollen grains, this is a best taxonomic significance. The main taxonomic significance in a plant kingdom region is due to presence of different designings in the exine of pollen grain. Due to presence of different designings in the pollen grain exine, it indicates taxonomic significance. These pollen grain or pollen grains are having different properties. For example, some of the species of pollen grains are rich of nutrients. Whenever the pollen grains are rich of nutrients, those pollen grains used to produce pollen products. Especially the pollen products usage very common and more in western countries like US and UK and other countries. Reason is the pollen grains are rich of micronutrients. In those countries, from the pollen grains, from the nutrients, the people can prepare pollen syrups and pollen tablets. In recent years, the usage of these pollen products such as pollen syrups and pollen tablets widely used in India also. Generally, if the people are using these pollen tablets and pollen syrups, it's a rich of nutrients and it's giving more energy, and the performance of horse races and athletics will be increases. Reason is these pollen grains are rich of nutrients. The next one, these pollen grains for future plant breeding program purpose. Some of the pollen grains are having disease resistant characters, maybe insect resistant and pest resistant. Some of the plant species pollen grains are having high productivity characters. Because of these characters, for future plant breeding program purpose, these pollen grains are preserved in liquid nitrogen medium at minus 196 degree centigrade. This phenomenon is called as pollen banks, is also called as cryopreservation. The preservation of desirable pollen grains at minus 196 degree centigrade in liquid nitrogen medium is called as cryopreservation or pollen banks. The pollen banks' main intention is to improve the plant breeding program. And one more. Some of the species of pollen grains are having allergic properties, especially if the pollen grains are dispersed by winds, that is called anemophilous. The dispersal of pollen grains by wind currents is called as anemophilous. In anemophilous plants or wind dispersed pollen grains, if these pollen grains are having allergic property, those allergic pollen grains are very microscopic. It measures 25 to 50 micrometer diameters. Then it can be easily enters into nasal cavity and reaches to lungs. And it causes respiratory disorders like asthma and bronchitis. Those pollen grains were called as pollen allergy. Some of the species of allergic pollen grains, Parthenium styroporus, it is commonly called as carrot grass. One more plant species, Chenopodium, and one more plant species, Amaranthus. In the plant of Parthenium styroporus, commonly called as carrot grass, Chenopodium plant species, Amaranthus plant species, these plant species pollen grains are having allergic property. They are causes respiratory disorders like asthma and bronchitis. Those pollen grains we called as pollen allergy. The next properties of pollen grains is pollen viability. Pollen viability means at mature condition of anther, when the pollen grains are released from the anther and these pollen grains land on surface of stigma by the process of pollination. So, release of pollen grains from the anther and land on surface of stigma, this in between this period pollen grains are having viability. The period, the simple definition for pollen viability, the period in which pollens have the ability to germinate on the surface of stigma. After release of pollen grains from the anther, if the pollen grains have ability to germinate on the surface of stigma, that period we called as pollen viability. The period in which pollen has the potentiality to germinate on the surface of stigma is called as pollen viability. The pollen viability is not constant for all species, it varies from one species to another species. For example, in the plants of wheat and rice, after release of pollen grains, if the pollen grain reach on same species surface of stigma within 30 minutes or half an hour, pollen germination can take place. If it is crosses after 30 minutes, there is no pollen germination. That means the pollen viability in the plants of wheat and rice is half an hour or 30 minutes. Whereas in the plants of Rosaceae and Leguminosae plant species, the viability of plants is, pollen grains is few hours. These are the different properties of pollen grains, such as pollen products, pollen banks or cryopreservation, pollen allergy and pollen viability. One very most important characteristic feature, one of the species of plant, Parthenium styroporus, commonly called as carrot grass. This plant species pollen grains are having allergic property. These plant pollen grains came into India with imported wheat. When the imported wheat contaminated with Parthenium pollen grains. This is the origin of Parthenium styroporus or carrot grass in India. The last pre-fertilization event during sexual reproduction for male gamete development. The topic is development of male gametophyte. For male gametophyte, the beginning cell or the starting cell is pollen grain. Pollen grain is also called as microspore. Generally, the characteristic features of pollen grain are microspore. These are unicellular, uninucleated, haploid, spherical shape measures 25 to 50 micrometers diameter, made up of 2 unit membranes, exine and intine. Exine is chemically made up of sporopollenin, intine is chemically made up of pectin plus cellulose. A thin area is present in the exine where the sporopollenin is absent is called germ pores. When this beginning cell or microspore or pollen grain for male gametophyte development, it undergoes first mitotic division. The haploid unicellular uninucleated pollen grain produces 2 unequal cells. The larger cell is called as vegetative cell and the smaller cell is called as generative cell. Due to first mitotic division, the unicellular haploid pollen grain or microspore undergoes first mitotic division to form 2 unequal cells. Larger cell is called as vegetative cell, smaller cell is called as generative cell. The shape of vegetative cell nucleus is irregular shape and large size. And the shape of nucleus, generative cell nucleus is spindle shape. So, the generative cell, it is float in the cytoplasm of vegetative cell. Now, this 2-celled pollen grain, the larger cell is called generative cell, smaller cell is called, the larger cell is called vegetative cell, smaller cell is called generative cell. Especially in 60% of angiosperms are in flowering plants or in advanced plants. The pollen grain shed from the anther at mature condition in 2-celled stage. In how much percentage of angiosperms pollen grains shed from the anther are released from the anther? If the question, then the answer is 2-celled stage or 2-celled pollen grains. Larger cell is called vegetative cell, smaller cell is called generative cell. In rest of 40% of angiosperms, the pollen grains release in 3-cell stage. One is vegetative cell, 2 male gametes. Now, these 2-celled pollen grains in 60% of angiosperms release from the anther and land on the surface of stigma. On the surface of stigma, when water and nutrients are available, the pollen grains outermost exine will rupture and intine forms pollen tube. Intine forms pollen tube. The pollen tube come outside of the pollen grain through the thin areas present in exine is called as germ pores. When the pollen tube is formed, the smaller cell nucleus, generative cell nucleus, spindle shaped, it's move into the pollen tube, it undergoes second mitotic division to produces 2 haploid amoeboid shaped male gametes are produced. Formation of 2 haploid amoeboid shaped male gametes in a pollen tube is nothing but male gametophyte development. The male gametophyte development beginning cell or starting cell is pollen grain or microspore. The question is, formation of 2 haploid male gametes in a pollen tube require how many mitotic division? Answer is 2 mitotic division. And one more question. For male gametophyte development or for male gametophyte, the mother cell is pollen grain or microspore? This is the second question. And the third question, which is the mother cell for male gametes? The mother cell for male gametes is generative cell or generative nucleus. The formation of 2 haploid male gametes in a pollen tube from microspore or pollen grain, it requires 2 mitotic division. One of the scientists, his name is Amici. He was the first scientist, first discovered formation of pollen tube from the intine. Pollen tube was first discovered by Amici scientist. If a pollen grain, one pollen grain produces one pollen tube. Such kind of condition is called as monosiphonous. If one pollen grain produces many pollen tubes, is called as polysiphonous. Poly means many, siphon means tube. One more question. In how much percentage of flowering plants are angiosperms? Pollen grains shed from the anther. The answer is two-celled stage or two-celled pollen grain. Name the two cells. Larger cell is called vegetative cell, smaller cell is called as generative cell. The shape of vegetative cell nucleus is irregular shape. Shape of nucleus is spindle shape, generative cell nucleus. This is one of the pre-fertilization event in this second chapter, formation of haploid male gametes. Male gamete formation is over. In the next class, we discuss how female gametes are formed. Megasporogenesis and female gamete formation and female gametophyte development we discuss in the next class.